Okay, let's unpack this. We are diving deep today into something absolutely fundamental happening inside all of us all the time. The cell's big decision. That choice between life, maybe reversible injury, and, well, death.
It really is a critical decision point.
Yeah.
And when people hear cell death, they usually picture, injury, maybe poison, something messy and accidental.
Right, the sort of catastrophic failure.
Exactly. But the reality is most cell death isn't like that at all. It's highly controlled. It's this molecularly orchestrated self-destruction.
And that's really our mission today, isn't it? To get under the hood of this, to understand the difference between that accidental, messy death, which we call necrosis,
Right.
and this other process, this regulated, precise, almost elegant form of cellular suicide known as apoptosis.
Right.
We need to go, way beyond just defining it and really dig into the molecular nuts and bolts.
Yeah, that distinction is absolutely key. Necrosis, like you said, it's messy. It's usually a result of some overwhelming injury, physical or toxic. The cell swells up, bursts open, spills everything out.
Causes a whole lot of trouble for its neighbors.
Precisely. Inflammation, damage. It's bad news. Apoptosis, though, is totally different. It's an active process triggered from within by this built-in suicide program. It uses specific enzymes, mostly these things called caspases.
We'll definitely be talking more about those.
Oh, yes. They go in and systematically dismantle the cell's insides. They degrade the DNA, the structural proteins. It's surgical.
Surgical precision. That's what makes it so fascinating, isn't it? It's deliberate, programmed, not just a breakdown. And the name itself, I find that interesting, too.
It is quite evocative, yeah. Apoptosis, it comes from ancient Greek. It means falling off, like leaves falling from a tree in autumn or petals from a flower.
Ah, so it implies something natural, scheduled almost.
Exactly, a kind of controlled shedding, something necessary. And interestingly, it was first recognized just by how it looked under the microscope, its morphology, way back in 1972. That was long before we really understood the complex molecular machinery driving it.
And the sheer scale of this. When you realize how many cells are doing this, the precision needed is just, well, staggering. It's not just a few cells here and there deciding to check out.
Not even close. We're talking about maintaining homeostasis. Keeping the right number of cells in every tissue, keeping everything stable and in balance.
Right.
And to do that, estimates suggest that in the human body, something like, get this, almost 1 million cells undergo apoptosis every single second.
1 million per second.
Wow. Yeah, it's this constant phenomenal turnover rate. And it happens silently, precisely, ensuring the whole organism stays stable without constant alarms going off.
Because if that many cells died messily through necrosis, 1 million explosions per second, we'd basically go walking inflammation crisis.
You absolutely would. Which brings us back to the outcome, the clean exit, which is really the defining feature of apoptosis at the tissue level. With necrosis, the key failure is that cell membrane rupture. Everything leaks out.
The 5-alarm fire you mentioned.
Exactly. Enzymes digesting neighbors, triggering this big inflammatory response. But apoptosis, it's the opposite. The cell tidies up first.
It packs its bags before it leaves.
That's a great way to put it. The dying cell shrinks down, then it fragments into these neat membrane-bound packages. We call them apoptotic bodies.
And the crucial part is the outer membrane stays intact.
That's the critical difference. Even as the membrane is blebbing and changing shape dramatically, it holds together. It keeps all those potentially damaging contents, the active enzymes, the DNA fragments, safely sealed inside these little packages.
So nothing leaks out into the neighborhood.
Correct. No leakage, no major inflammatory signal. It's like a controlled demolition instead of an explosion. The debris is neatly contained.
Followed by, presumably, very efficient trash removal. A silent, clean process designed purely to protect the surrounding tissue. Okay, let's shift gears a bit. Let's talk about why. Why do cells activate this internal kill switch? Because like you said, most of the time this isn't about disease. It's actually essential for normal life.
Absolutely. Apoptosis is fundamental. It's there to eliminate cells that are, well, no longer needed or maybe damaged beyond repair or simply present in excess numbers. It's all about maintaining that tissue balance and function.
And probably the most famous example, one that really coined the term programmed cell death, is during development, right, in the embryo.
That's the classic example. While apoptosis is the mechanism of death, the term programmed cell death really refers specifically to its role during embryonic development and tissue remodeling. It's basically the body's sculptor.
Sculptor? How so?
Well, it removes supernumerary cells that were needed temporarily during development but now have to go. Think about how your hands and feet form. Initially, there's tissue between the digits, like webbing.
Ah.
Apoptosis is what removes those cells, precisely carving out the fingers and toes. Without it, we'd keep that webbing. It's essential for shaping organs and removing structures that are only needed early on.
It's amazing that the same basic process shaping us in the womb is still working away decades later to keep us healthy. How does it operate in adults under normal, everyday conditions?
In adults, you see apoptosis very clearly linked to adaptation, especially with hormones. We call this hormone-dependent involution. Think about tissues that change cyclically.
Like the lining of the uterus.
Exactly. The breakdown of the endometrial lining during the menstrual cycle when progesterone levels fall, that's driven by apoptosis. It's a scheduled removal.
So the body is resizing, retooling itself based on these hormonal cues.
Precisely. You also see it in the regression of breast tissue after lactation stops, or in the ovaries during menopause as follicles undergo atresia. These are large-scale removals of cells triggered by hormone withdrawal. And beyond hormones, it's crucial for maintaining turnover in tissues that proliferate rapidly.
Like the gut lining you mentioned earlier.
Yes, the intestinal crypt epithelial cells. They turn over incredibly fast, replacing themselves every few days. The older cells at the tips of the villi get pushed off, and they die cleanly via apoptosis. If that didn't happen efficiently, you'd risk growths or impaired nutrient absorption.
And the immune system, with all those cells being produced, it must rely heavily on apoptosis just for regulation, right? To prevent chaos.
Oh, absolutely. This is one of its most critical protective roles: immune regulation and establishing self-tolerance. Your body makes billions of immune cells, T cells and B cells.
Right.
But during their development, many are generated with receptors that could mistakenly attack your own healthy tissues, self-antigens.
Autoimmune potential.
Exactly. If those self-reactive lymphocytes were allowed to mature and circulate, they'd cause autoimmune diseases. Apoptosis acts as the crucial quality control step, primarily in the bone marrow and thymus.
So it's like an internal security check, eliminating the dangerous recruits before they even get deployed.
That's a perfect analogy. It weeds out the potential traitors. And furthermore, apoptosis is essential for cleaning up after an immune response. Once an infection or inflammation is dealt with, you have all these extra immune cells hanging around neutrophils from the initial fight, lymphocytes from the specific response.
They serve their purpose, but now they need to go.
Right. They die via apoptosis and are quickly cleared away by macrophages, allowing the tissue to return to its normal, quiet state without residual inflammation.
That quick, non-inflammatory cleanup seems vital. So that covers the sort of healthy physiological side of apoptosis. But it also plays a huge role as a defense mechanism when things go wrong pathologically.
Yes, when cells are injured beyond their capacity to repair, or if essential survival signals disappear, apoptosis is often triggered as a protective measure. It's often the lesser of two evils, preventing something worse like cancer or widespread tissue damage from a necrotic cell bursting.
And the prime example here must be DNA damage, right? From things like radiation, chemo drugs, even just free radicals.
Absolutely central. If a cell's DNA gets severely damaged, its internal surveillance systems kick in. If the repair mechanisms can't fix it, the cell faces a choice. Try risky repair or self-destruct.
And it often chooses self-destruct.
Often, yes, especially if the damage is extensive. This decision is heavily influenced by a key protein, the tumor suppressor P53. It senses the damage. If P53 determines the damage is irreparable, it initiates the apoptotic cascade.
That's the ultimate sacrifice, isn't it?
Yeah.
The cell kills itself to protect the organism from the risk of it becoming cancerous due to those mutations. It's a built-in anti-cancer system.
It is fundamentally an anti-cancer mechanism. Eliminating these potentially mutated cells prevents them from proliferating and forming a tumor. And conversely, when this apoptotic pathway fails, maybe P53 is mutated or downstream signals are blocked. That's a major step towards cancer development.
We also see apoptosis kicking in when there are problems inside the cell's protein factory, the endoplasmic reticulum.
Yes, this is known as endoplasmic reticulum, ER stress. The ER's job is folding proteins correctly. If misfolded proteins start piling up, maybe due to a genetic defect or just overwhelming stress, the cell senses this quality control failure.
Like a backlog on the assembly line.
Exactly. It first tries to fix the problem with something called the unfolded protein response, or UPR. But if the UPR can't clear the logjam, if the stress persists, the cell triggers apoptosis to eliminate itself, preventing the toxic buildup of bad proteins. This is thought to play a role in diseases like cystic fibrosis and some neurodegenerative conditions.
And pathology often involves infections, too. Apoptosis seems deeply involved in how we fight off viruses.
Definitely. It's a two-way street with infections. Sometimes the virus itself actually triggers apoptosis in the host cell HIV, and adenovirus can do this, but more often it's a crucial part of the host's defense.
You mean our immune cells inducing apoptosis?
Yes, specifically the cytotoxic T lymphocytes, or CTLs. These are the immune system's assassins. They recognize cells that are infected with viruses or tumor cells, and they directly induce apoptosis in those target cells.
A targeted kill order.
Precisely. It eliminates the source of the infection or the dangerous tumor cell. It's incredibly effective.
Sounds brilliant, but potentially damaging, too.
That's the downside. While killing infected cells is necessary if the infection is widespread, the sheer amount of CTL-induced apoptosis can cause significant tissue damage itself. Think about severe viral hepatitis.
The liver damage isn't just the virus. It's also our own immune system killing off infected liver cells.
A large part of it, yes. The CPLs are doing their job eliminating the virus reservoirs, but the massive wave of hepatocyte apoptosis, a massive challenge. It's a delicate balancing act. Block apoptosis too much, and you risk chronic infection. Let it run unchecked in a severe infection, and you risk organ damage. Fine-tuning that response is a major goal.
And one last pathological role, pathologic atrophy. This is where an organ shrinks due to cell loss.
Right. This can happen, for instance, if a duct in an organ like the pancreas or a salivary gland or the kidney gets blocked. The cells downstream are starved of signals or damaged by pressure buildup.
And they eventually give up and trigger apoptosis.
Exactly. The gradual loss of these parenchymal cells via apoptosis leads to the shrinking, the atrophy, and the loss of function in that organ.
Okay, so we've covered the why, the normal functions, and the roles in disease. Now let's get visual. If you're a pathologist looking down a microscope, how do you spot apoptosis, especially if it's meant to be so quick and clean?
The morphologic changes are actually quite distinctive, quite different from necrosis. They are the original defining features, really.
And it starts with the cell's overall size, you said, unlike necrosis where things swell up.
Exactly. Apoptotic cells do the opposite. They undergo dramatic cell shrinkage. The cell pulls inwards. The cytoplasm becomes very dense, often staining brighter pink eosinophilic because the water gets squeezed out and all the internal organelles get tightly packed together.
Okay, so shrunken and dense. But the real giveaway is in the nucleus.
That's the most characteristic feature, definitely. Chromatin condensation. The DNA, the chromatin, clumps together. It forms these dense, often crescent-shaped masses right up against the inside edge of the nuclear membrane.
Like it's packing everything up neatly before demolition.
That's a good visual. It really looks like the cell is compacting its genetic material. Following that, the nucleus itself fragments. This involves pinosis, which is the nucleus shrinking down, and then karyorrhexis, where it actually breaks into pieces.
And the DNA inside is getting chopped up too.
Yes, that condensed chromatin is being actively cleaved by enzymes into specific sizes, usually into fragments the size of nucleosomes. Seeing that fragmented nucleus is a very strong indicator of apoptosis.
And all this internal breakdown leads to the final packaging step.
Right. The cell membrane, which has remained intact so far, starts to bulge outwards, forming these protrusions called blebs. Then the whole cell breaks apart into these membrane-enclosed fragments, the apoptotic bodies.
Containing bits of cytoplasm, organelles, those nuclear fragments, all neatly wrapped up.
Perfectly sealed, yes. These are the tidy packages ready for disposal.
But you mentioned it's hard to spot sometimes because it's so fast.
That's the challenge. The whole process, from initial trigger to fragmentation, can be quite rapid. And crucially, these apoptotic bodies are usually cleared away by phagocytic cells, like macrophages, within minutes.
So you might not see fields of dying cells, just maybe an isolated apoptotic body being swallowed.
Exactly. Or you might just see slightly increased cellularity as macrophages move in to clean up. The key negative finding is the absence of inflammation. If you see dying cells but no inflammatory cells rushing in, that strongly suggests apoptosis rather than necrosis.
It's a remarkably efficient, almost stealthy process. Which brings us neatly to the engine driving it all, the molecular machinery, moving from what we see to how it actually happens. And you said this whole thing hinges on a specific family of enzymes.
Yes, the caspases, they're absolutely central. The name itself tells you their function.
They are cysteine proteases that cleave target proteins specifically after aspartic acid residues, hence C-aspases.
Got it. And they're the executioners.
They are the ultimate executioners, yes. But they don't just float around active. They exist initially as inactive precursors called proenzymes or procaspases.
So they need to be switched on.
Precisely. They need to be cleaved themselves by other proteases or sometimes each other to become active. Finding active caspases in a cell is basically the biochemical confirmation that apoptosis is happening. It's a cascade effect.
A chain reaction. And you mentioned two main phases.
Broadly, yes. There's the initiation phase. This is where the first caspases in the chain, the initiator caspases like caspase 8, caspase 9, caspase 10, become activated in response to a death signal. They start the process. And
then comes the execution phase. The initiator caspases activate the downstream executioner caspases, primarily caspase 3 and caspase 6. These are the real workhorses that go out and dismantle the cell by cleaving hundreds of different target proteins.
And critically, there are two main ways to kick off that initiation phase, two main pathways, but they eventually converge on the same executioners.
That's right. We have the mitochondrial pathway, also called the intrinsic pathway, and the death receptor pathway, or extrinsic pathway. They start differently, but they both ultimately lead to the activation of caspase 3 and caspase 6.
Okay, let's tackle the intrinsic pathway first, the mitochondrial one. This seems to be the main route for apoptosis caused by internal problems.
It is. It's the major mechanism triggered by things like DNA damage, protein misfolding, lack of essential growth factors or survival signals, or even certain developmental cues. It's the cell-sensing internal distress.
And it all comes down to the mitochondria, the cell's powerhouses, making a life-or-death decision.
Essentially, yes. The critical event is an increase in the permeability of the mitochondrial outer membrane. When this barrier is breached, specific pro-apoptotic proteins leak out from the space between the inner and outer membranes into the main body of the cell, the cytoplasm.
And the key player leaking out is cytochrome C. Which is weird, because isn't that essential for making energy?
It is. That's the incredible duality. Inside the mitochondrion, bound to the inner membrane, cytochrome C is absolutely vital for the electron transport chain, for cellular respiration. It keeps the cell alive.
But if it escapes?
If it leaks out into the cytosol, its function flips completely. It becomes a potent trigger for the apoptotic cascade, a real Jekyll and Hyde molecule.
So what controls whether cytochrome C stays put or leaks out? What guards that mitochondrial gate?
That control lies with a crucial family of proteins,
Yeah.
the BCL2 family. They are the ultimate regulators, the gatekeepers of the mitochondrial outer membrane. They exist in this constant state of balance, deciding whether the membrane stays intact or becomes permeable.
And this family has different members, some promoting survival, some promoting death.
Exactly. We can group them based on their structure, these things called BH domains, and their function. First, you have the anti-apoptotic members. Primarily BCL2 itself, BCL-XL, and MCL1.
These are the good guys, trying to keep the cell alive.
From the cell's perspective, yes. They typically reside in the mitochondrial outer membrane, and ER and cytosol too, and their job is to maintain that membrane's impermeability. They prevent BX and BA, the pro-death proteins, from poking holes in the membrane. Survival signals, like growth factors, often work by boosting the levels or activity of these anti-apoptotic BCL2 proteins.
Okay, so BCL2 and friends are holding the line. Who are the attackers trying to breach it?
Those are the main pro-apoptotic members. BX and BIC. When they get activated, they change shape, come together, oligomerize, and insert themselves into the mitochondrial outer membrane. They essentially form channels or pores.
Ah, so BX and BA create the holes that let cytochrome C escape.
That's the current thinking, yes. They directly permeabilize the membrane. So the fate of the cell really hangs in the balance. Is there enough active BCL2, BCL-XL to block BX to bake, or do BX-BAC overcome the defenses?
It's like a molecular tug of war at the mitochondrial surface. But what tips the balance? What tells BX and BAC to attack, or DCL2 to stand down?
That's the role of the third group within the family. The regulated initiators, often called the BH3-only proteins. Think of names like BD, BIM, BID, PUMA, NOXA. These are the sensors.
Sensors for what?
Sensors for cellular stress and damage, lack of survival signals, DNA damage, ER stress. These conditions lead to the activation or increased production of specific BH3-only proteins.
And what do these BH3-only sensors do once they're activated?
They act in two main ways to ensure the pro-death signal wins. Firstly, some of them can directly bind to and activate BX and BEG, telling them to form those pores. Secondly, they can bind to and inhibit the anti-apoptotic proteins BCL2 and BCL-XL, effectively neutralizing the cell's defenses.
So they either activate the attackers or disable the defenders, or both. A double whammy.
Exactly. It ensures a robust switch towards permeabilization once a critical stress threshold is reached.
Okay, so the BH3 sensors tip the balance. BAX, BAK form pores, and cytochrome C floods into the cytosol. What happens next? How does cytochrome C actually start the caspase cascade?
Once in the cytosol, cytochrome C finds and binds to another protein called APAF1, apoptosis activating factor 1. This binding triggers APAF1 molecules to cluster together, forming a large, wheel-shaped complex. This structure is called the apoptosome.
The apoptosome sounds ominous.
It is, sometimes called the wheel of death. Its crucial job is to recruit molecules of pro-caspase 9, the key initiator caspase for the intrinsic pathway.
So the apoptosome gathers the inactive caspase 9.
It gathers them and facilitates their activation. Within the apoptosome complex, the pro-caspase 9 molecules are brought close enough together that they can cleave and activate each other through autocatalysis.
And active caspase 9 then does what?
Active caspase 9 then turns around and activates the downstream executioner caspases, particularly caspase 3, and boom, the execution phase is fully underway.
It's such an intricate series of checks and balances, but there's one more layer, right? Something about inhibitors.
Yes, there's a final failsafe. Cells normally contain proteins called IAPs, inhibitors of apoptosis proteins that can bind to and neutralize executioner caspases, acting as a brake.
Okay.
But when the mitochondria become permeable, they don't just release cytochrome C, they also release other proteins, like SMAC Diablo. And the job of SMAC Diablo is to bind to the IAPs and prevent them from inhibiting the caspases.
Wow. So it releases the trigger, cytochrome C, and simultaneously releases the safety catch by neutralizing IAPs. It really commits.
It really does. Once that mitochondrial outer membrane goes, the pathway is designed to proceed very rapidly and irreversibly.
Okay, that's the intrinsic pathway sensing trouble from within. Now, what about the extrinsic pathway, the death receptor one? This sounds more like the cell receiving an external kill order.
That's a good way to think about it. This pathway is initiated by signals from outside the cell, typically delivered by other cells, often immune cells. It involves specific receptors on the target cell's plasma membrane.
Death receptors.
Right. These are members of the TNF receptor superfamily, and the key feature is that they have an intracellular portion called a death domain. This domain is essential for relaying the signal inside once the receptor is activated.
And the classic example involves a receptor called FAS.
Yes. The FAS system, also known as CD95, is the best characterized. The FAS receptor is present on many cell types. Its activator, its ligand, is called FAS ligand, FAS-L. FAS-L is typically found on the surface of activated T lymphocytes, especially those cytotoxic T lymphocytes, CTLs we talked about.
So when a CTL wants to kill a target cell, like a virus-infected cell,
it uses its FAS-Ls to bind to the FAS receptors on the target cell surface.
And what does that binding achieve?
The binding causes multiple FAS receptor molecules to cluster together in the membrane. This clustering brings their intracellular death domains close together,
creating a landing platform inside the cell.
Exactly. This platform of clustered death domains then recruits adapter proteins. The key adapter here is called FADD, FAS-associated death domain.
FADD binds to the clustered death domains.
Correct. And FADD, in turn, has another domain that recruits inactive procaspase 8 and sometimes procaspase 10. This whole assembly, the clustered FAS receptors, FADD, and procaspase 8, is called the DISC, the death-inducing signaling complex.
The DISC, so it forms right there at the membrane.
Yes. And the critical function of the DISC is, again, proximity-induced activation. By bringing multiple procaspase 8 molecules very close together, it allows them to cleave and activate each other.
Generating active caspase 8.
Generating active caspase, which is the key initiator caspase for the extrinsic pathway. Active caspase 8 can then directly cleave and activate the executioner caspases, like caspase 3, triggering cell death.
Now, given how powerful this is, especially for the immune system killing unwanted cells, surely there are ways to block it. Viruses must have figured this out.
Oh, absolutely. The evolutionary arms race is very evident here. Many viruses, and even our own normal cells sometimes, produce a protein called FLIP. Yes. FLP is structurally similar to procaspase 8 and can insert itself into the DISC. However, it lacks the enzymatic activity. So it binds, but it can't be activated, and it blocks the recruitment or activation of the real procaspase 8.
It's a decoy. It jammed the DIC mechanism.
Precisely. It's a very effective way for viruses to protect their host cell from being killed off too early by CTLs, buying more time for replication.
Fascinating. But you mentioned earlier these pathways aren't totally separate. How does the extrinsic signal link to the intrinsic mitochondrial pathway?
There's a critical link, especially important in some cell types like liver cells or pancreatic beta cells. Active caspase-8, generated by the DISC, can actually cleave one of those BH3-only proteins we discussed earlier, specifically a protein called BD.
Ah, cleaving BD activates it.
Yes. Cleaved B, often called TBID for truncated BID, then translocates to the mitochondria and acts just like the other stress sensor BH3-only proteins. It activates BX and BK.
So the external death signal via caspase-8 and BID essentially triggers the internal mitochondrial pathway as well.
Correct. It feeds the signal into the intrinsic pathway, leading to cytochrome C release and caspase-9 activation. This serves as an amplification loop, ensuring that even if the direct activation of executioners by caspase-8 is weak, the much more powerful mitochondrial pathway is engaged to guarantee cell death. It makes the kill signal much more robust.
Okay, so whether it starts intrinsically via stress and cytochrome C release, or extrinsically via death receptors, we end up with active executioner caspases, caspase-3 and caspase-6. They're now fully unleashed. What specific damage do they do to finalize the demolition?
They are proteases, so they cut proteins. They have hundreds of targets within the cell. Some of the most critical actions include, well, remember that, DNA fragmentation.
So those nucleus-sized pieces, yeah.
The executioner caspases make that happen indirectly. They cleave a protein that normally inhibits a DNA cutting enzyme, a DNase. This inhibitor is called ICAD inhibitor of caspase-activated DNase.
So they cut the inhibitor,
which releases the brake on the DNase. The DNase, called SAD, then becomes active, gets into the nucleus, and starts chopping up the DNA between the nucleosomes, creating that characteristic ladder pattern you see on a gel.
That explains the nuclear breakdown perfectly. What else do they cut?
They go after structural components. They cleave proteins in the nuclear matrix, which helps the nucleus itself to fragment. They cleave proteins in the cytoskeleton, which likely contributes to the cell shrinkage and the membrane blebbing we see. They basically dismantle the cell's internal scaffolding and information storage,
leading to that final formation of the neat apoptotic bodies.
Exactly. The precise mechanics of bleb and body formation are still being worked out, but the widespread proteolysis by executioner caspases is clearly the driving force behind the cell's physical disassembly.
Right. The cell has now been dismantled and neatly packaged. The final crucial step for keeping things clean is the disposal. You called this efferocytosis.
Efferocytosis, yes. It's the specific term for the phagocytosis, the engulfment and digestion of apoptotic cells or bodies by phagocytes, usually macrophages, but sometimes other neighboring cells too.
And this isn't just passive cleanup. The dying cell actively signals for pickup.
It absolutely does. It sends out signals. The most famous are the eat me signals.
Eat me signals. Seriously.
Well, that's the functional term. The best-known one involves a lipid called phosphatidylserine, or PS. Normally, PS is kept strictly on the inner leaflet of the cell membrane.
Hidden away inside.
Right. But during apoptosis, enzymes called flipases and scramblases cause PS to flip to the outer surface of the membrane. This exposed PS is a direct signal recognized by receptors on phagocytes.
So it's like the dying cell puts up a flag saying, "Take me, I'm ready."
That's exactly it. It's a very clear signal. In addition to eat me signals like PS, the dying cells also release soluble find me signals, chemoattractants that actively draw phagocytes to the site.
Making sure help arrives quickly.
Ensuring rapid clearance, yes. And the speed and efficiency are key. Because efferocytosis normally happens before the apoptotic body loses its membrane integrity completely, the dangerous contents stay contained, the phagocyte engulfs the whole package.
Preventing that messy leakage you get in necrosis.
Precisely. And there's another crucial aspect. When a macrophage engulfs an apoptotic cell via efferocytosis, it doesn't trigger inflammation. In fact, it does the opposite.
It dampens inflammation.
Yes.
Yeah.
The process actively suppresses the production of pro-inflammatory cytokines by the macrophage. It promotes the release of anti-inflammatory signals. So the cleanup is not just silent, it's actively anti-inflammatory.
That is incredibly elegant. The whole process, from the internal decision to the final silent cleanup, is designed to minimize disruption to the surrounding tissue.
Absolutely. So if we synthesize this, the core idea is that apoptosis is this incredibly sophisticated, molecularly controlled pathway. It's designed to eliminate cells that are unnecessary, unwanted, or dangerously damaged, but to do so cleanly, silently, without triggering the damaging inflammation associated with necrosis.
And understanding this process, especially the balance points like the BCL2 family at the mitochondria or the caspase cascade, that knowledge is fundamental for understanding and potentially treating so many diseases.
Really essential. Think about cancer. A hallmark of cancer is often the evasion of apoptosis. Tumor cells figure out ways to shut down this suicide program.
Like overproducing BCL2, that anti-apoptotic guard.
Exactly. Overexpressing BCL2 or BCL-XL, or mutating p53 so it can't sense DNA damage, or having viruses like HPV produce proteins that degrade p53. These are all strategies cancer cells use to gain immortality by resisting apoptosis. Developing drugs that reactivate apoptosis in cancer cells is a major therapeutic goal.
And on the flip side, too much apoptosis is also a problem.
A huge problem. In many neurodegenerative diseases, Alzheimer's, Parkinson's, Huntington's, the core issue is the inappropriate premature death of essential neurons via apoptosis. Stroke damage also involves excessive apoptosis in the surrounding, initially viable tissue.
So there, the goal would be to inhibit apoptosis to keep those crucial cells alive longer.
Precisely. Finding ways to selectively block the apoptotic cascade in neurons, for instance, without causing other problems, is a key area of research for those conditions. It's this constant balancing act.
It really highlights how apoptosis is this double-edged sword, essential for health, but devastating when it goes wrong in either direction.
Life and death controlled at the molecular level.
Which leads perfectly into our final thought, something for you, the listener, to mull over. We mentioned that staggering number. Almost 1 million cells dying via apoptosis in your body every second.
Just think about that relentless, continuous turnover. It means that those molecular arbiters we discussed, proteins like BCL2 holding the line, BAX and BAK ready to attack, they have to maintain an exquisite balance, constantly, for your entire life.
So here's the question. What happens if that balance flickers just slightly? If, say, BCL2 gets a tiny temporary advantage in a few cells, letting them survive a stress signal they should have died from, or if BXBK become just a fraction too sensitive.
Consider the cumulative effect of tiny imbalances over decades. How finely tuned must this system be to prevent catastrophic tissue loss from too much death, or the runaway cell survival that leads to cancer from too little death?
Think about the sheer complexity and the demand for near-perfect regulation on those BCL2 family proteins, acting as the gatekeepers of life and death for trillions of cells 1 million times a second. It's perhaps the most profound example of molecular quality control in our biology.
And when that control falters, even slightly, the consequences can be immense. It really underscores the precarious beauty of cellular life.
And with that fascinating, maybe slightly unnerving thought, our deep dive into apoptosis comes to a close.